Chapter 3, Segment 4, has 9 parts to it, the first being receptor molecules recognizing transmitters. Otto Lowery designed an experiment that would definitively discriminate between two candidate modes of transmission, chemical versus electrical. In this experiment, he stimulated the vagus nerve of one frog, which caused it to slow its heart rate down. Then he collected a sample of fluid surrounding the frog's heart. He then took that sample and bathed a second frog's heart with it. And this then caused the second frog's heart to slow down, which in turn helped scientists to learn that the nervous system uses chemical signals. Nicotinic cholinergic receptors are found at synapses on muscles. Most of them are excitatory, whereas some are inhibitory, and then there are also some receptors that are both excitatory and inhibitory. The nicotinic acetylcholine receptor resembles a lopsided dumbbell with a tube running down its central axis. There are many different kinds of receptors and which show regular variation. An increase in receptor number is referred to as upregulation, and then a process that decreases receptor density is called downregulation of that receptor. The next section is transmitters bind to receptors, gating ion channels. So the first definition is ionotropic receptors, which directly control ion channels. When bound by the transmitter, the ion opens and flows directly across the membrane. Another definition to note is metabotropic receptors, which recognize the synaptic transmitter, but do not directly control the ion channels. G proteins are also convenient because they bind to the compound guanosine diphosphate and guanosine triphosphate, and it plays an important role in transmitting and binding receptors. If we think of the neurotransmitter as an external message that is arriving at the receptor site, the next chemical signal activated inside that cell would be the second messenger. So we have the first messenger, and then the second messenger is also important to know. The second messenger simply amplifies the effect of the first messenger. 80% of the known transmitters and hormones activate cellular signal mechanisms to recept coupled G proteins. There are two basic units to G proteins. One important one to note is the alpha subunit, and it migrates away within the cell and modulate the activity of its target cell. The next section, being the action of synaptic transmitters, is stopped readily. Timing of synaptic transmission is necessary in many neural systems to drive the rapid cycles that may occur. There are two important things to note in this. One being degradation, which is the rapid breakdown of a transmitter into inactive metabolites, and two being the reuptake, the process by which released synaptic transmitter molecules are taken up and refused by the presynaptic neuron. And within the reuptake, there are transporters, which specialized receptors in the presynaptic membrane recognize transmitter molecules and return them to the presynaptic neuron for reuse. The next section is several factors regulate neurotransmitter release. Through autoreceptors, the presynaptic cell is informed about the net connections of neurotransmitters in the synaptic cleft and may regulate future neurotransmitters release to adjust that concentration. There are about 4 types of chemical transmissions that should be noted. The first being axon dendritic, the second being axon somatic, third being axon-axon, and the fourth being dendrodendritic. So it's just basically where it's traveling to. So axon to dendrite, dendrite to dendrite, so on and so forth. Then there's another important definition to know, retrograde synapse transmission, which starts with classic axodendritic synapse activity, but subsequently releases a gas neurotransmitter, and this could be something like CO2. The next section being neurons and synapses combine to make circuits. So the nervous system is comprised of many different neural circuits, and each circuit has basic functions like cognition, emotion, and action. And so it's important to note that they're all interconnected with one another some way, shape, or form. An example of this would be the knee-jerk reflex, so that's all connected throughout our body. And the name for this would be a neural chain, a straightforward linking of neurons. The next section is cross-electrical activity of the brain is readily detected. So detecting electrical activity in the brain, essentially. Gross brain potentials are divided into two different classes, one being ones that appear spontaneous without any stimulation, and two being a particular electrical activity that was stimulated. A recording of a spontaneous brain potential or brain wave is also called an electroencephalograph, or commonly known as an EEG. The next section is seizure disorders result from electrical storms in the brain. Seizures are manifested by electrical character of the nervous system, so extensive connections among nerve cells generate massive waves of intense nerve cell activity that seem to involve almost the entire brain. We can also refer to this as epilepsy, as we commonly know is a disorder involving seizures. One important definition to know is tonic-clonic, which is a type of generalized epileptic seizure in which the nerve cells fire in high-frequency bursts. Another definition being simple partial seizures, a seizure that is characterized by spike and wave EEGs and often involves a loss of awareness and inability to recall events surrounding the seizure, so like loss of consciousness and things like that. The third type of seizure that's also important to know is complex partial seizure, a type of seizure that does not involve the entire brain and therefore causes a wide variety of symptoms. And this could just happen in like a certain part of the brain, like the frontal lobe, so on and so forth. And then there's something called kindling that's also important to know. It's not a type of seizure, it's more of a method that researchers use to study seizures, and it's mainly used in animals. And this is a method of experimentally introducing an epileptic seizure by repeated stimulation of a brain region. Another section being event-related potential is measured changes resulting from discrete stimuli. So an unaware stimuli just stimulating the brain in unknown ways. So gross potential changes evoked by discrete sensory stimuli, such as light flashes or clicks, are often called event-related potentials. So the light switching on and off, that's considered an event-related potential. Computers are being used to record these potentials using electrodes. It's also important to note that a decrease in the amplitude of certain waves or increases in the lightning latency have been valuable for detection of hearing impairment in young children. So we're using the technology in computers and electrodes to test for deafness in infants, and we're seeing the brain activity through this technology. Lastly, optogenetics. So optogenetics uses genetic tools to insert light-sensitive ions ion channels into neurons so that stimulating the brain with light-delivered fiber optic cables can excite or inhibit targeted neurons. So that just means we're using genetic tools to alter the neurons in a way that we can detect certain things. And there are two specific examples the book gives, and one being the channel hordospin and the halodorspin. So these two types are important to know as you continue your reading. Thank you.